Hello, everybody, and welcome to Mr. Saucedo's YouTube videos. Today, we're going to be talking about replication of DNA. So, how does DNA get replicated? During our talk about the cell cycle, when we were looking at mitosis, we kind of said, hey, DNA it makes a copy of itself. We'll deal with those details later on. Well, it is now later on. So, it's time to go over the details of replication. Replication is the process of copying DNA prior to a cell dividing. And remember, that happens during the S stage, during interphase, right here. That is when your DNA is making a copy of itself. And so, why? Well, it ensures that when your cell divides, both cells have a complete and accurate set of DNA after mitosis is done. Remember that there's a critical checkpoint at G2 before your cell divides, and that if it detects any sort of irregularity in your DNA, your cell is programmed to die, and we call that apoptosis. So there are three main steps to DNA replication. The first step is called initiation, and there's an enzyme called helicase. You can probably guess what it does. It unwinds your double helix. So remember, helicase ends in an ace, it's an enzyme. And helic is referring to the helix part, right? And so it basically goes right down the middle of your DNA and breaks all of those hydrogen bonds as it goes down, like a pair of scissors cutting through wrapping paper or something. It is just like undoing a zipper, okay? There's also an enzyme called primase, which adds primer pieces of RNA in order to help with the next step. So primase is not adding DNA, it's adding little RNA primers to help with our second step in DNA replication. So the next step is called elongation. So there are an entire class of enzymes called DNA polymerases. And so DNA polymerase will attach to the primer and it'll start to pair things up, just like you are pairing up cards or something. It'll say, okay, there's a C, G needs to go there. Okay, there's an a T needs to go there and pair it up that way. And so that's what it's doing as it moves down the unzipped piece of DNA. And then the last thing is termination. So termination, you have to remove those primers that we added, and now you should have two complete sets of DNA. And you even have a proofreading mechanism. So DNA polymerase and another enzyme called nuclease proofread the work to make sure that there are limited errors. And again, that doesn't mean that it's perfect, okay? But it does get rid of a lot of mistakes that would have been made if it wasn't proofread before it was allowed to move on to the next step. Because then we just have a whole bunch of cells getting popped all the time. So you want limited errors. If it notices that, like, a T is accidentally paired up with a C, it'll rip out the T, and then it'll have to, it'll get replaced by the appropriate nucleotide. Sorry, I almost said subunit. Okay, how long does DNA replication take? It only takes a few hours, okay? And the two strands each have one original strand, and that's called the template strand. And also, remember, these enzymes are going through, and so we also have one new strand that's called a complement. And so you can see it here. We started with this blue double helix. Helicase is breaking those bonds, and then we are literally, the reason why DNA replication is so good is because the original template strand doesn't get damaged along the way. All that's happening is it's saying, okay, there's an A here, that must mean I add a T here. Okay, there's a C here, I must add a G here. And so you have a complete and accurate record of what was there, and then you are just trying to fill in the blanks. If you're these little, polymerases here, you're just trying to fill in the blanks. And so we call DNA replication a semi-conservative process because the DNA strands contain one template and one complement. Now, keep in mind, enzymes are very particular. DNA has a direction to it. So you can think of it like this. The time has come to say goodbye. Okay, there we go. That would be like my original message. And I am pairing that up and I'm making a complement to it. Okay. So in reality, it has to be completely conserved. If this were not a semi-conservative process, I would just say, okay, the time has come to say goodbye. Okay. And then I would copy it letter for letter. And I wouldn't have damaged this at all. So that would be a plus. That would be a completely conservative copy, right? But also you're a little bit more prone to errors. So instead, what we do is something like this. We get half the message, okay? The other enzyme gets the other half. And then we just fill it in with whatever's supposed to come, like that. So this is actually the semi-conservative process. This is a completely conservative process where the original strand is untouched and we are just copying and making something completely new. The reason why the semi-conservative process is a little bit more, I don't want to say it's better, but it's a little bit safer is because you still have the original half. And so all you're doing is filling in and trying to pair up the other part to that. Okay. So I just said before we went over that DNA has a direction. And so the direction we like to call uh, based on it's based on a prime. Okay. So the complement strand is facing what we would call 5, and that's not an apostrophe, it's a prime is what we would call that apostrophe. So it's facing 5 prime to 3 prime. It is constructed continuously because it is going the direction that it is supposed to be going, okay? And so you can see this top one here, this little RNA primer was dropped off by primase, helicase unzipped it already. And so our enzyme is able to just kind of go 5 prime to 3 prime, and it's able to continuously make it. Now, the opposite one, though, is going 3 prime to 5 prime. It's going the opposite direction. And so it is constructed in little pieces. And so it is discontinuously copied. And we call these little fragments here Okazaki fragments, okay? And I know this sounds weird, but the piece of DNA that is being made from 5 to 3 is getting made a lot quicker and faster than the DNA that is being copied that is moving 3 prime to 5 prime. And that's because it's literally the enzymes are reading everything backwards. And so here we go. Read that. Please read that. All right, okay. But what if instead, if you're wondering why do this? Like why Okazaki fragments? Well, what if instead we're only reading small bits of it? It becomes a lot easier. There was a cat that's at on a mat. That's why Okazaki fragments are utilized, because since they're doing everything backwards, the other enzymes that are moving up, they're a lot more prone to errors. And so you want to make sure that you're not making any mistakes along the way. Okay. Now, remember these enzymes that are moving, these polymerases, DNA polymerase can't reach the ends of our DNA. They can't. And remember, the ends of our DNA are protected by little caps called telomeres. And so that means that every time it reaches the end of your strand of DNA, a little bit of genetic material is lost because DNA polymerase can't get all the way to the end. It has to end early and clip off a little bit. And so that's why we keep junk DNA at the ends of our, and at the beginning of our chromosomes, is because the little bits of it are going to get lost along the way. So if we had any important genes there, that would not be very advantageous to us. Now, proofreading occurs only when a mismatch is detected. So DNA polymerase and nuclease move back one nucleotide and remove it. So if it notices it, it'll say, oh, wait, got to go back, and it'll go back and it'll clip it off and it'll get replaced. This is what it actually looks like. And I think it looks super cool, by the way. But this is what that process actually looks like. Not in real time or anything, right? But it is like a molecular machine, which is super cool. And so it is literally saying, okay, great, built that part, going down the list, going down the list. Let's pair them up, let's pair them up. And it's pretty good. All right, so
Replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions, and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
and all of that happens every time your cell divides. So if we look at the following template strand, so let's pretend like we are DNA polymerase here. This is what I'm given. What would be the complementary strand that I would build from this? Well, remember, A always pairs with T, C always pairs with G, G always pairs with C, T always pairs with A, right? So I can fill in the rest as I go. And just like that, you just replicated a piece of DNA.